Okay, great. I wanted to first thank the symposium organizers for inviting me. This has really been a lot of fun for me. I loved hearing all of the cool new science yesterday and getting to chat with many of you. So what I'm gonna do today is tell you a little bit about some recent interactions between mobile genetic elements, specifically plasmids and phages, that we have, for lack of a better word, just sort of stumbled upon as we were working to develop an environmentally relevant model system for looking at host virus interactions in heterotrophic marine bacteria. So I think it's fair to say that bacteria can harbor really a diverse array of mobile genetic elements, some of which are shown here on this slide from a review article. Things such as plasmids, viruses, phages, transposons, genomic islands, and so forth. And we know that mobile genetic elements are critical in driving bacterial evolution and influencing genetic diversity in natural populations. We also know, and this is becoming an increasingly sort of popular area of study, that within an individual microbial cell, that multiple mobile genetic elements can interact with one another. And we are fortunate enough yesterday to hear about a really elegant example of a lytic phage and a genomic island interaction that occurs in Vibrio cholera. And that interaction is really important in the evolutionary arms race between that bacterium and its lytic virus. We also had the opportunity to hear some really compelling evidence indicating phage plasmid and phage interactions occurring in natural populations that was evident from using high-C technology. And one of the real advantages of that type of approach is that we can begin to get insight into those types of mobile genetic element interactions in natural populations without requiring culturing of those organisms. And so as I had alluded to earlier, really what my focus is going to be on today is interactions between temperate phages and plasmids within a single bacterial host. And so this slide is the details of which are unnecessary, I think, for this audience, but I included it to remind you that when it comes to temperate phage, the paradigm is that these viruses can exist principally in one of two states, one where they're acting lytically, other when they can integrate into the genome of their host, and they can replicate alongside their host replication until which sometime that they actually are induced and the lytic genes are turned on and they initiate the lytic cycle. Most of what we know about temperate phage lifestyles comes from studies of bacteria of human and agricultural importance, with good reason. And the paradigm that has emerged from those studies is that it's host stress, principally, that can be a principal driver of prophage induction. And so the long-standing analogy, and one which I think is a good one, is that temperate viruses will remain as prophage as long as the bacterial host is growing under conditions that are conducive to it growing, dividing, replicating happily. But once it undergoes some sort of a stress and initiates a global stress response, is the most common or well understood mechanism, that the so-called SOS response, that you get this induction. So there have been a lot of benefits in the context of the host that have been discovered from strains that are lysogenic, that is, that harbor prophage within their genomes. And the one that I wanted to really highlight here, which is on the far right, is one that maybe is not one that people think too much about. And that is when lysogenic bacteria, through low levels, so-called spontaneous induction, i.e. that would be induction without obvious and overt external stressors, are releasing, those prophage initiate the lytic cycle. And so a limited number of those cells lyse, releasing phage into the environment. And if those if those phage are capable of attacking susceptible competitors, that's a mechanism whereby a subset of one population is sacrificed to the larger benefit of the rest of the population. And I'm going to come back to that. That's why I highlighted that. So when probably one of the things I should have led with, though I think I did, I said I'm interested in studying heterotrophic marine bacteria. I'm a marine microbial ecologist. I got into studying viruses in an atypical way. I essentially had a student who came to me several years ago and said he was interested in studying viruses that infect the group of bacteria that I study. And I said, that sounds great, but I don't know anything about viruses. And he said, that's fine. And so in retrospect, I think I owe a lot of what our lab has been able to do in the last couple of years to his original determination to isolate and study viruses that infect this group of bacteria, which I'll tell you more about in a minute. But one of the things that I wanted to highlight was the prevalence of lysogeny in the oceans. So in about 2016, there was a study that was done looking at the prevalence of prophage in closed bacterial genomes. And the result of that analysis was that 50%, approximately 50% of bacterial genomes, closed genomes, so that was going to be a little bit of a biased view of the bacterial world, had prophage. And so we wanted to look to see how pervasive that might be in marine populations. And so I had a student who took, he didn't depend on having closed genomes. He was willing to look at draft genomes that were available in JGI. He restricted his search to organisms that come from the marine environment. And he, in addition to using complete or draft genomes, he also looked at metagenome assembled genomes, as well as single cell genomes as well. And so what this is effectively showing is that the phyla that he looked at, he restricted his analysis to those phyla for which he had at least 10 representatives. And then using what we felt was the most appropriate and conservative prophage detector, which was Viersorter, which was developed by Simon Roux. He then went through and tried to catalog which of these genomes looked like they had evidence for prophage. And so what you can see from these analyses, so they're color coded depending on whether they are in this first column, whether their complete genomes are in this reddish color, and if they're draft genomes, they're in this greenish color. Light colors mean no evidence for prophage. The darker color indicates an evidence, evidence for prophage. And so to make a long story short, he found evidence for prophage, certainly not as high as 50%, but again, this was a less biased viewpoint of bacterial genomes in the oceans. And we fully recognize that we are likely missing some because we did not require or restrict ourselves to only looking at closed genomes. And so one of the groups of marine bacteria that we're interested in is the so-called Rosibacter clade. And so organisms within this group are extremely abundant and ubiquitously distributed across the world's oceans. They can be isolated from sea surface waters as well as hydrothermal vent systems. And I was excited to see in Leila's poster yesterday where she was looking at viruses in deep sea hydrothermal vent systems, that she saw evidence for Rosibacter viruses down there as well. We find them associated with sea ice. They're very abundant in coastal systems, including coastal salt marshes of the southeastern US, which is an area where my group does a lot of work, and also associated with marine snow and marine particles. Rosibacters are also well known to be associated with marine eukaryotes, particularly photosynthetic eukaryotes, including algae and phytoplankton, including the Coccolithophormelian Huxleyi. And when we look at the genome sequences of the various Rosibacters that have been sequenced, along with physiological studies, we see that these organisms collectively have adaptations that enable them to or facilitate their lifestyle with both living and dead primary producers in marine systems. So I had alluded to earlier on that I had a graduate student who came to me and was interested in studying viruses of rosibacters. And so in order to do that, he went out to an Aemiliana huxleyi phytoplankton bloom. It was a mesocosm experiment that was being done in a fjord in Bergen, Norway. And he isolated many different bacterial strains and viruses that infect them. And so the one that I want to tell you about today is a strain that's called Cephalobacter pontiacus, and we call it strain CBD. We recently ended up closing the genome for this organism, and the reason that we did that is because it was really important for our work to understand what the plasmid content of this particular strain was. And so this is just showing a graphic of the genome of this organism. So it has a main chromosome that's about 3.3 megabases, but then it has 4 large, very low copy plasmids that it maintains in its cell. And so these 4 plasmids range in size from 72 kb to 176 or almost 77 kb. There's a tremendous amount of genetic material that's encoded on these plasmids. They all contain unique toxin antitoxin systems that are supposedly involved in plasmid maintenance. So these are these post-segregational toxin antitoxin systems. The idea is that, well, first of all, let me just back up and say, when I say very low copy number, these plasmids in an individual cell, you'll have on average 1 to, in one case, plasmid 3, up to 5 copies of the plasmid in an individual cell. So when you have these low copy plasmids, typically they'll have these toxin antitoxin systems on them in order to try to force the strain to maintain these plasmids, because it's very easy during cell division that not each of the daughter cells retains a plasmid. So the way that these TA systems work is you have a long-lived toxin and a relatively short-lived antidote to that toxin. So if you end up with a cell that doesn't have this plasmid associated with it, it effectively dies because of the presence of the residual toxin in the absence of the antidote. So one of the things, in addition to having 4 plasmids, one of the things that's of interest to us is that this strain, that when it was isolated from nature, is lysogenized. And so it has a 42 kb prophage that resides within its genome. And when my graduate student Charles used this strain to isolate viruses that infect it by plaque assay, he isolated 3 different viruses, one of which I want to focus on today is another temperate virus. And it has a tremendous amount of sequence variation to the prophage that resides within the bacterial genome. And so this is just showing a sequence similarity between the prophage on the bottom and then this temperate virus that was isolated by plaque assay from the same waters that the bacterium was isolated from on the top. And so I always like to say that these 2 viruses are nearly identical, except for the 2 regions where they're not. And so those are these 2 regions here, one which we think is important in determining receptor specificity, and then the second, which we believe is important in dictating the lysogenic lytic switch, because there's a number of transcriptional regulators. And so in the regions where these 2 viruses are similar, they share nearly 100% identity, and in the regions where they're not identical, they really are distinct. One of the things that we noticed when we had the sequence for this virus was that it has this putative integration site, that it shares the putative integration site with the prophage within the original bacterial host. And so again, we isolated this virus by plaque assay, so it will initiate a lytic infection on its host. But we also determined that if you infect this host with this virus, that you can get a number of outcomes. One of which is you get a lytic infection and production of that virus. And so I should mention, I'm going to try to use color coding here. We'll just refer to these as the red and the blue virus. But you get a lytic infection. You also get induction of the resident prophage. So you get production of the alternate phage genotype. We don't get complete lysis of the population. So we get cells that survive and retain the original prophage. And then we also get something that we think is interesting, which is we get, for lack of a better word, we get, it looks like a replacement of the blue virus with the red virus in the host. We also noticed that, at least in some cases, when you infect this strain, actually in the original experiment where we isolated a host strain that had the alternate phage genotype, that it also had lost 2 of the 4 low copy number plasmids. Despite the high degree of sequence similarity, these viruses do not offer immunity to the opposite genotype, right? They're susceptible to infection by the opposite genotype. We see evidence for homoimmunity. If you infect the blue lysogen with the blue virus, you don't see any indication of infection, and you don't see indication of induction. Alternatively, if you add the red virus to it, you get an active lytic infection, and again, also induction of the blue. And then the same is true if you flip that dynamic. If you take the blue virus and infect the red host, you get a lytic infection, but you don't get a lytic infection if you add red to red. So one of the things that was interesting to us is despite this high degree of sequence similarity, again, because we think that there's differences, that we think that what's driving this is differences in the transcriptional regulation that controls the lysogenic to lytic switch. We basically have these 2 viruses that are in competition with one another. And so we had done, let me skip ahead and show the results, and then I'll come back to that. We had done some just sort of head-to-head competition experiments to ask sort of who would win if you took a bacterium that had the blue virus versus a bacterium that had the red virus. And we asked what the outcomes would be depending on the growth state of the bacterial host. And part of the reason that we did that, so we tested it in liquid culture, but we also did it as biofilm. And part of the reason that we did that is because Rhosiobacters and Sulfidobacters in particular are really well known to sort of transition really readily between a free swimming as well as a biofilm lifestyle, a surface associated lifestyle, the so-called swim versus stick phenotype, and switching between those 2. And so let me just start off by saying, if you grow the red lysogen in liquid culture, monoliquid culture, and you can compare it to growth of the blue lysogen in monoliquid culture, we see that the red lysogen doesn't grow to the same cell densities as the blue lysogen. When we do a biofilm assay for each of these 2 in monoculture, we see that you get a far more robust biofilm when you have the red lysogen compared to the blue lysogen. And then when we do these competition studies where we're adding these strains at a 1-to-1 ratio, we see some interesting dynamics. We see the competition in liquid culture doesn't reach the same final cell density as the blue in monoculture. And then when we look at sort of who would win, and I should say this is after 24 hours, we see that there are far more red bacteria that survive and more red viruses that are produced. If we look at this competition, again, a head-to-head competition in a biofilm, we see a much more robust biofilm, and we see that it appears that the blue phenotype beats the red phenotype after 24 hours. And so one of the things that is important to mention is, again, this red lysogen is missing two plasmids. And this red lysogen also has what we realized is higher rates of what we're calling spontaneous prophage induction. So again, in the absence of any obvious inducer, you're just growing the spectrum under sort of normal lab conditions, it spits off a high number of infectious particles. And when we're thinking about putting these two organisms in direct competition with one another, because you're starting with this one red virus, that even at time zero, where we removed any extraneous virus, we've just added these two cells, this red virus is already starting to throw off virus that initiates a response by the blue lysogen, right? It initiates the induction of the blue prophage. And so you get this warfare that's starting to occur. And so that's one of the things I wanted to sort of bring back to this original example that I had shown you earlier, where you have one population that's using its viruses to gain a competitive advantage over another population. And what we're seeing with our two-phage one-host system is that you actually have pitted a host against itself, depending on what virus is integrated into its genome. And you see evidence of this reciprocal attack. And so getting back to this idea of spontaneous induction, I just wanted to take a minute, though I think some people were showing these types of assays yesterday, so I think this should be pretty straightforward. But when I'm talking about spontaneous induction, I just want to make sure you understand what I'm saying. So basically, you have a bacterium that has a lysogen, you grow it in liquid culture, for all intents purposes, you think it should be happy. You let it grow for a period of time, after which, or even during which, you can take samples, you can remove the cells, so you just have the viruses, and then basically you can assay to see how many infectious particles do I have in this specific experiment. And the way that we do that is that we basically are doing a plaque assay on the alternate prophage genotype, which is the appropriate permissive host. And so, as I had alluded to, so we saw this real distinction in terms of spontaneous induction in our blue prophage containing strain. When we would grow it in liquid culture, we would never see evidence of free virus. And that was in contrast to the red virus containing strain, in which we would see evidence of infectious virus being produced. And again, we'd also see that those cultures never reach the same cell densities as their blue lysogen counterparts. Now, one of the things that we were trying to deduce is the distinction between the two due to the prophage genotype, or is it due to the plasmid, the host genetic background in terms of the plasmid content? And so one of the things that we started to do was to essentially try to generate additional derivative strains that had different prophage genotypes. And so what I'm showing here is the original parent strain. It's been infected with the A virus, and then each one of these is the offspring. And A indicates that this is now the opposite prophage type. And then over here on the right, we're also looking at the plasmid content in each of these strains. So we started with a strain that had all the plasmids. After the superinfection experiment, where we got conversion to the alternate prophage type, in one of these strains, plasmid 1 was lost. In this other set of experiment, when we started with a strain that had the A prophage type and was already missing plasmids 2 and 4, we then reverted it back to the D prophage type, and then looked at the plasmid content, and we had an additional strain that lost an additional plasmid, this plasmid 1. And then what I'm showing over here on the left-hand side is the evidence for spontaneous prophage induction in each of these strains. And so I hope that you can see the takeaway, which is in any case where plasmids are missing, the strain shows evidence of prophage induction, regardless of the genotype. And this relationship is not restricted to one plasmid type. You see it with plasmid 1, plasmid 2 and 4 in combination, or 1, 2, and 4 together, which we thought was interesting, to say the least. Because we saw evidence of plasmid 1 and 2 and 4 getting lost, we asked a very simple question, which is, well, how stable are these plasmids in routine culturing of these organisms? One of the things that I failed to mention, but probably should mention, is that we were the ones who isolated this original bacterial strain about 10 years ago. And we've always been very careful. It went through 3 passages, and then it went straight into the freezer. And so whenever we're working with this strain, we never keep it out past 3 to 4 passages. After it's been through 3 or 4 passages, we go back to the original freezer stock. So we tried to minimize domestication of this strain, right? And so, but one of the questions that we asked, because plasmid loss is a frequent response to domestication of bacteria within labs. And so one of the things that we simply asked was, what if we just kept passaging these bacteria? Do any of these plasmids begin to go away? And so we did this for strains that contained our original strains that contained all 4 plasmids at time 0 and one prophage genotype, and then the other that was missing 2 plasmids. And to make a long story short, this was just done over 20 passages, and we saw that the plasmid that was most frequently lost was plasmid 1, that large plasmid. We have this slide is showing results from 20 passages. We've now taken that out to 50 passages, and the long and short of it is that we can get combinations of plasmids to be lost if you just keep passaging these bacteria. But we see a trend, which is that in almost all cases, it's usually plasmid 1 that goes first, and then additional plasmids are lost. And so we really began to sort of focus on plasmid 1 because it was the one that seems like it's lost most readily. It's also the biggest of the plasmids, and it contains the singular flagellar gene cluster, the only flagellar gene cluster encoded within the genome. And so when these bacteria lose this plasmid, they also lose the ability to produce a flagellum and swim. And so one of the things that we were interested in was, again, sort of trying to understand what the role of this plasmid might be in interactions with prophage. And in our attempts to characterize a strain that was just missing plasmid 1 and comparing it to a lysogen that had plasmid 1, we noticed that regardless of which prophage genotype, if you add the alternate virus type, you can get a lytic infection. But if plasmid 1 is missing, that cell now seems to be immune to infection by the alternate genotype. Yet it also has a relatively high rate of spontaneous induction. And so we did absorption assays and basically just asked, do we see evidence for this virus to be able to absorb to cells that have or are lacking plasmid 1? And it seems that we don't see evidence of absorption, nor do we see evidence of production of, in this particular case, the blue virus when you add the blue virus to the strain. So we don't see evidence of absorption, nor do we see evidence of lytic infection. But again, we do see evidence of prophage induction in that strain. So we've got some interesting interactions. So The presence of plasmids seems to be important for stabilization of these prophage, regardless of the genotype. We don't know what that mechanism is. It's one thing we're trying to work out. And then we're also seeing that one plasmid in particular plays a pretty important role in susceptibility of the host to infection. One of the things that we wanted to look at as well is distribution of the genes associated with these different plasmids, as well as these two viruses. And so this is just showing a summary of what these distributions are from the Terra Ocean survey. And we found that we see evidence for these plasmids mostly at subphotic zone levels, which might be consistent with the lifestyle of the bacterial hosts that house them. So just to sort of wrap it up, so we have some evidence for plasmid-dependent stabilization of prophages and also plasmid-dependent susceptibility to infection. And I am out of time, so I'll just quickly thank everyone who's done this work and the funding agencies, and thank you for your attention.